And based on the results, you implement changes if they're warranted. So the first step is really to know what you're trying to achieve. So, for example, in the case of viable antibiotic alternatives, how do we assess whether or not those products are actually useful under field conditions? And the approach I'm talking about can really apply to just about any new ingredient or evaluating management changes or equipment or so on. But I'm going to focus on the area of antibiotic alternatives. So if we look at traditional feed supplements like a phytase versus removing inorganic phosphate, our questions can be what alternatives will achieve the same performance at the same or lower cost, or what will improve performance? But when we look at antimicrobial alternatives, it's a little bit more complicated because usually to have a successful strategy, we need multiple products and different approaches, different mechanisms in order to replace that single product. And so if you look at the experience globally, it's not possible to take antibiotics out of the feed and replace that with a single product. Usually it's a combination of different products with different mechanisms. And of course, that means there are going to be higher costs. So really, the question then becomes, will I be able to sell my product or not? And depending on the country you're in, that can be due to legislative restrictions. That can be due to the demands of export markets. But it can also be related to consumer preferences. So will our consumer continue to buy our product if we continue using antibiotic growth promoters? And so the way we approach the question is a little bit different than a strict economic or performance evaluation. So in order to assess antibiotics, we really need to look at what antibiotic growth promoters are actually doing. And really, they're growth permitting antibiotics. They're allowing the bird to get closer to their genetic potential. They're not allowing the bird to achieve growth beyond their genetic potential. So when we evaluate, we have to ask what is a reasonable expectation of antibiotic growth promoters, and then what's a reasonable expectation of antibiotic alternatives. So, for example, if the genetic capacity or the genetic potential for growth is a certain level, this is usually our positive control in an experiment. And then we want to have a negative control, an appropriate negative control, where we see the effects of an actual challenge, so reduced performance, indicating that our, in this case, positive control or antibiotic is having a positive effect. Now, we apply our various treatments, and hopefully we're getting at least a partial recovery of performance. We may get a complete recovery of performance, in which case we may be able to evaluate that product or that alternative strategy as being as effective under those experimental conditions as the positive control as the antibiotic. Now, in rare circumstances, but we shouldn't necessarily expect this, in rare circumstances, we may find our alternate strategy performs better under our challenge conditions than the positive control. But again, we're looking at antibiotic alternatives, antibiotic replacements. And so usually the best that we can hope for is a complete replacement or a complete recovery of effect under challenge conditions. So again, we have to temper our expectations when we look at evaluating the outcome of our experiments. So how do we do that? Well, first of all, we need to ask a clear research question. We have to have a good, solid hypothesis that can be tested. We want to have a limited number of variables, and we want to have good experimental design. So wherever possible, and I'll talk about this in field trials, wherever possible, we want to have both a positive control where we're evaluating our existing strategy, antibiotics, and a negative control where we remove the antibiotics. And we want to have some sort of challenge present. It doesn't necessarily need to be an applied disease challenge, but we certainly want to have conditions where the antibiotics actually have something to do and show an effect. And so we need to show that, first of all, the antibiotics are actually providing protection or stimulating growth and what have you. And then we can apply our treatments and compare the relative effects of recovery of performance. We need to make sure we have good replication of treatments. We want to block appropriately, and we want to take appropriate samples. So many of us are familiar with university scale experiments. These are typically small scale, intensive, highly controlled research facilities, and very often they are facilities that may or may not reflect the real world. And so we use these university experiments, and I do these all the time, to test ideas. Do the ideas work? Are they feasible? But we recognize that these are not real world conditions, and so the results may not apply directly to field research. So if we're looking at transitioning from research results to field application, then we need to make the next step, which is, do the research ideas tested at the university work under my conditions as a producer or as a poultry company? So there's a number of different ways that we can do that. We can make changes and then compare the results with historical data from the same facilities. So this is called benchmarking. Or we can test on multiple commercial farms, either over time or using a large number of farms. We can test results. Some companies have their own research farms to help make this transition. And then there are also there's an approach of using small research pens within commercial barns and commercial flocks. So how do we conduct field research? Well, again, first we have to ask the right question. We want to be specific. So we don't want a vague question of what happens if we change some things. We really want to focus on, for example, what's the effect on feed conversion ratio when we use this ingredient in place of antibiotics? We generally want to change one thing or one variable or as few as possible at a time so that we can really focus on what's the effect of that change. So I mentioned benchmarking. Benchmarking is a basic approach. We record data from our facilities over time, and we look at trends over time. So we understand what the performance was under our previous conditions or our standard conditions. We make our change, and then we compare the performance under the new conditions. And again, this can be over multiple farms. It can be multiple cycles through the same farm. And then we can evaluate our results and decide whether we want to implement the change or make further studies or reject the change. So some advantages, this could be a very simple approach. It's using data that a company has often already collected. The company actually needs to use that data effectively. And the advantage is that this really reflects the reality of your day-to-day operations. Now, there are also some challenges. It can be difficult to make only one change at a time. In commercial production, of course, there are changes in feed ingredients and supply all the time. Environmental conditions can change over time, the chick supply, and there's so many different things that can influence the outcome of an experiment. So we want to make sure that as much as possible, we're minimizing variability and increasing repeatability. So making sure that we're taking into account the effective season and housing type, geography and weather, and so on. Again, we want to objectively evaluate the results. So we want to have a way to statistically evaluate the output, the data from our experiments. Usually we have to do this over multiple flocks, and so that can be over a long period of time, or again, in many different barns at the same time. And we need to have good quality records. And in commercial production, obviously, there are other things that the company might change in terms of barn modifications, the season, the changing genetics, and so on. So there are some limitations to a benchmarking approach. Now, if we look at designing experiments and carrying them out in commercial farms, we can use multiple commercial farms. We can repeat this in time. We can have a positive control, but it's very often difficult to have a negative control because we don't want to have a complete loss of protection that antibiotics would give us. We might be limited in the number of experimental treatments. So we need to make sure that we're able to replicate, either replicating treatments on individual farms or replicating treatments across multiple farms, or ideally, we would have all treatments represented on the same farm, but have that repeated multiple times. So this allows us comparison of different treatments under real world conditions. We can be more confident in our results perhaps than with benchmarking. And depending on the number of farms involved, we can conduct our experiments in a relatively short period of time. Again, there are some challenges. So this is more complex to plan and conduct than simply looking at benchmarking. It requires usually the participation of many different levels of the company, from executives to managers to the actual people in the barns doing the work. And so sometimes in commercial production, we can see that differences in between farms can actually be greater than differences between treatments. The next approach is to look at company-owned research farms. So this is something like a cross between commercial production and university facilities where we have a smaller scale commercial unit that's subdivided into pens. Again, this allows us replication, this allows us blocking, this allows us within the same environment to assess multiple different treatments. An advantage is that it allows for company-specific management and feeding programs and veterinary programs and so on. So it really reflects directly to what a particular company is doing. So again, we can have controlled conditions to a certain extent. We can have positive control. We're more likely to be able to have a negative control, although again, this is difficult. And we can use our experimental treatments. And the advantage is that we have replication of treatments under the same environmental conditions. So we have a lot more control over variables. It requires a lot fewer workers than company-wide studies where we've got many, many different farms and many different conditions, and it allows us to be more consistent. So it allows producers, it allows companies to test new ideas on a small scale before expanding to their entire operation. So their feed, their management, and their processing plant all are an important part of this research. This can be challenging. Again, just like taking university results and applying them to large scale, this is closer to the real world, and so companies can be a little more confident in the results, but it's still a different environment than a commercial barn. And you still need to have multiple levels of the company involved. And if you've got a good research team, if you've got buy-in from all levels of the company, this can be a very successful approach. And then another interesting approach is to use research pens within your commercial barns. So you can set up smaller pens within the actual production environment. Again, you can certainly have your positive control. It may be difficult to have a true negative control with no protection, but you can also have your multiple experimental treatments, and this allows replication of treatments on individual farms or even within individual barns. And so these are close to practical conditions. And again, it allows you greater replication of treatments within commercially relevant environments. So there are some challenges. It requires modification of facilities. You have to change feeder lines, water lines, in order to provide feed and water to the pens. You can see a picture here, supplied with its own feeder and its own water line, and a small subpopulation of the birds. You need to make sure that your pens are properly set up so that birds don't escape or enter either from the general population or from another experimental pen. This can be a lot more labor intensive within that barn because those pens need to be managed individually somewhat. And then there's always the question of how big should the pens be in order to be commercially or provide a relevant environment for the birds. And also how many pens per treatment, making sure good replication. So however we do our experiments under commercial conditions or semi-commercial conditions, we then need to evaluate the results. And so we need to be aware of the value of the results, but also the limitations. Again, doing field research can be challenging because of some of the variables that are introduced just by nature of doing field research. So if we're doing a single study, we need to be aware that variations in response, either due to what we see at university trials versus what we see in commercial application. Or if we're doing commercial studies, what happens when we're looking at one location to another? There can be very, very different facilities, even within the same company. So that can influence our outcome. And again, we need to remember what is a reasonable outcome to expect from a study. So again, what we want to see is our our positive control performing well. We want to see that there is in fact a challenge, if possible, under commercial field conditions. Again, it can be very difficult to actually impose a negative control group or treatment where we don't have protection against intestinal disease, for example. And then we want to evaluate our different treatments and look, do we get performance similar to or equal to our positive control? And again, it may be unreasonable to expect greater performance. If we're replacing antibiotics, we want to make sure that we're getting equivalent performance. But antibiotics are very effective, so it's not likely that we're going to see additional performance or additional responses beyond what we see with antibiotics. So again, some challenges, particularly related to negative controls, and in the context of antibiotic-free production or tests, that means that there really is no protection against the pathogens that might be reducing performance or impairing bird health. And under commercial conditions, it's almost impossible to do that because it's an unacceptable risk to have no protection against bacterial diseases. There are production realities. There may be a disease outbreak that it has nothing to do with antibiotics. It may be a viral disease or a disease that is not controlled by antibiotics. And the company needs to be able to respond and change treatment. So that can influence the outcome of our experiments. It requires a buy-in from the staff. The staff has to pay a lot of attention to detail because we need to have good control. In my experience, sometimes when things are busy, or when something doesn't go exactly right, the production outcomes take priority over the research outcomes, and you can lose quality and control over your experiments very quickly. Again, you have to often have a large number of steps and a large number of people involved. So, for example, getting the right feed to the right bin or to the right barn. And so in our experiments, we often use colored feed bags or tags or bin labels to make sure that our experimental treatments are being applied appropriately. And then we need to make sure that there's a clear chain of command for maintaining the experimental protocol. So what happens when a mistake is made? What happens when someone changes things? How do we recover from that? How do we maintain the integrity of our research trial? So the next step then is to implement the findings. So based on the results, are the changes that we've investigated warranted? Does it make sense to apply it across the broader country? And really finding out what works and what doesn't. And then making that economic decision. Is it economically feasible? So is the cost of implementation worth it? Now, again, when we remove a single product like an antibiotic growth promoter, we're reducing costs by a little bit. I don't have time to get into the field of antibiotic alternatives, but typically what we see, the effective strategies are strategies that use different products, multiple products. And every time we add a new product or an additional product to the diet, we're adding cost. And so we need to look at the cost of protection, the cost of providing this, often what ends up being an insurance policy, and making sure that it fits into the economics of production. And then the final step is just ask the next question. So if we've had a successful outcome, we've made a decision. That decision might be to use a product or to not use a product, but we can be confident in our results. But then the next step is always ask the next question because we want to continually improve. We want to make sure that we're getting better and better. So in summary, our research in whatever form, whether it's university scale or commercial scale, we need to be doing research in order to continuously improve. And we need to recognize that no research approach is perfect, but will the outcome help to make the operation better? So we need to understand the opportunities, the limitations, and the type of research that we're doing in order to be able to implement and answer those questions. And if we design the experiments properly, we can ask good research questions, we'll have good experimental design and execution, and we'll be able to achieve an overall objective evaluation of potential alternatives to antibiotics. So thank you very much, and I'm happy to take any questions if there are any.
Thank you very much, Professor Dr. Dokover, for such an excellent presentation and wonderful insights. We completely understand your schedule and greatly appreciate the time you have spent with us today.
My pleasure.
If anybody have a question, please add your question in the chat box, or you can also ask directly to our keynote speaker just by unmute yourself. Professor Cover, please accept our sincere gratitude for your contribution to the GRC 2026. We will share your certificate of appreciation and conference material with you via the email afterward.
My pleasure.
Thank you very much. So we now move to our next keynote speaker. Professor Dr. Barn Kaspers, Professor of Animal Physiology at Ludwig Maximilian University of Monash, Germany. So let me give a very brief introduction of Professor Dr. Barn Kaspers. Professor Dr. Barn Kasper is a distinguished expert in avian immunology and animal physiology at Ludwig Maximilian University of Monash, Germany. With more than 3 decades of research experience, his work focuses on the innate and adaptive immune systems of poultry, host-pathogen interactions, mucosal immunity, and immune system development in avian species. Following his postdoctoral studies in veterinary medicine at the University of Monash, he completed postdoctoral research at the United States Department of Agriculture, USDA, USA. Professor Kaspers has authored more than 115 peer-reviewed scientific publications and serves as senior editor of the internationally recognized textbook Avian Immunology, 3rd edition. His research has significantly contributed to advancing knowledge in poultry diseases, including Marek's disease, avian influenza, and salmonella infections. Professor Kaspers has received several prestigious honors, including the Pfizer Award for Excellence in Poultry Research and the Marek Award for Achievement in Poultry Science, recognizing his global impact in poultry immunology and health research. So without further ado, I would like to invite Professor Dr. Van Kaspers to please carry on with your presentation. It's over to you, Professor.
Yeah, Sayam, thank you very much for the kind introduction, and I would also like to thank the organizers for inviting me to talk a little bit about our work or more recent work we are doing. Let me see that. Let me share my presentation. Okay, here we go. I go back. Get started. It's a bit slow. Okay, here we go. Do you see the presentation now?
Yeah, we can see your presentation.
Okay, excellent.
Perfect.
So again, thank you very much for inviting me. And as the previous speaker mentioned, I'm happy if you would share the presentation amongst your graduate students, but please don't share it on social media. There are still some data which are not published yet. Okay, so we will talk about the gut immune system in chickens and how it develops and what is the impact of microbial colonization on the development of the gut immune system. If we look at the immune organs in birds, we all know that T lymphocytes derived from the thymus, and we have those 8 lobes of thymic tissue on either side of the throat. We also have a very unique organ, the bursa fabricis. We are all familiar with that they produce B lymphocytes, which eventually will give rise to antibodies. And finally, we have the bone marrow, where maybe after bursa involution, B cells may be maintained and still produced. But there are also the heterophils, natural killer cells, monocytes produced in the bone marrow. And in particular, monocytes are an interesting population of cells because once they have left the bone marrow, they can disseminate in the body via bloodstream, and then they can differentiate in the tissue either into dendritic cells, which are professional antigen-presenting cells, or into macrophages. And after maturation of all these cells, they will leave the primary lymphoid organs and move via bloodstream to the secondary lymphoid organs. And here I've depicted the most important sites. Let's get started with the spleen. Everybody is aware of the spleen as a very important immunological organ. We also have organized lymphoid structures in the lung, which is called bronchus-associated lymphoid structures or tissues. You see these dark blue structures here all along the primary bronchus. So they get very early in contact with all particles and antigens which are delivered via airflow during breathing. And then we have the Hadarian gland, which is actually a lacrimal gland producing tear fluid, but half of the gland is also a lymphoid structure, as you can see here. And in the gut, we have a lot of different structures I will discuss in a moment. And all along the reproductive tract, we find lymphoid tissue and also, in particular, cells of the immune system. So if we focus on the digestive tract, we find these organized structures all along the gastrointestinal tract, starting with some tonsils up here in the pharyngeal area. We find a very distinct tonsil at the esophagus, again, another one at the pylorus. And then we have that Meckel's diverticulum, most of you are familiar with, I guess. We find pious patches and most prominent, these cecal tonsils at the junction of the ileum and the ceca in the birds on both sides. And finally, there's a bursa fabricis. Again, this is initially a primary immune organ generating the B lymphocytes, which will eventually home to different structures in the gut. And this is a picture depicting the bursa fabricis, nice round sac with an open duct to the cloacae. So in principle, indeed, content from the cloacae can be sucked up into the bursa fabricis and can actually be delivered to these lymphocytes inside the bursa fabricis. And then you see a single follicle with a distinct cortex and medulla. And I'm sure you all are aware of this structure because it's heavily disrupted during Gumboro infection. Here you can see nicely the cecal tonsils on either side, but we should be aware that there are immune cells all along the gastrointestinal tract, and not only in the ceca, but all along these tissues. A cross section of such a cecal tonsil, you see all these brown areas here. Each very small dot here represents a B lymphocyte. It's specific staining for B lymphocytes. And you can see here these organized structures called germinal centers, which are very important in producing good antibodies in response to vaccination or infection. But as I mentioned, throughout the gut, we have lots and lots of immune cells sitting in the lamina propria. You can nicely see here one single villus. See the crypts, and you can see that there are many, many immune cells sitting in the being part of the villus, and in particular sitting in the lamina propria. And this picture here just shows a single type of cells. It's just a picture of macrophages. And I would like to emphasize that there are also T cells, B cells, and in the context of infection, there will be more cells homing into the tissue, coming from the blood and going into this tissue. So the question is, how does the system develop? And I'm often asked, when is it fully functional? So if we look at macrophages first, these are pictures from broiler gut, 3 days, 7, 14, 21 days after hatch. And you can see that there's already a huge number of macrophages sitting in the lamina propria right after hatch. This system is basically functional at hatch. And the macrophages can phagocytose pathogens or foreign particles and eliminate those very efficiently. And we do see some change, but not all that much as the bird matures. So it's rather premature at hatch. But this system is also highly responsive. If you look at a 2 days old bird, you can see here, and this is a cross section of the cecum. And you see here macrophages sitting all over the place. But if you infect those birds with Salmonella enteritidis, you can see this huge influx of macrophages into the tissue. And a pathologist would right away say that is inflammation. There's a strong inflammation going on, and that's correct. And the question is, what is attracting those cells into this tissue? And there are a lot of chemoattractants produced in response to infection. They are collectively called chemokines, and chemokines are little proteins which diffuse from the place of production and then form a gradient, and macrophages will crawl along the gradient and then home into the tissue. And you can see here one example of a chemokine. It's not yet published. We are not entirely sure whether it's indeed CCL16, but we assume it is. But what is important to see, 2 hours after infection and 6 hours after infection, we don't see much of the cytokine expressed. But if you look at this, it's 100-fold increase in the production of this chemokine within 24 hours after infection, and that stays quite high for an extended period of time, actually. Okay, what about the other cells? Let's have a look at B lymphocytes, and this is in stark contrast to the macrophages, where we at hatch have a lot of cells, but B cells are basically not there. And that is also true on day 7, and it's still true 2 weeks into the bird. Very few B lymphocytes sitting here in the gut tissue. And it takes 2 to 3 weeks for those cells to show up in decent numbers and to start forming these organized structures. And these cells produce immunoglobulins, and in the gut, the most important immunoglobulin would be IgA. And if you look at IgA production, if we have no B cells, we have no production. And only at 14 days after hatch, or within the second week after hatch, the few B cells start to produce immunoglobulin A and help to protect from infection. And that really takes some time to mature. So while the innate immune system seems to be very mature at hatch, the adaptive immune system, at least for the B cell compartment, really takes time to mature in the gut. And that is also true for the T cell system, actually. If you look at these figures, they are very, very similar to what we've seen before for the B cells. Whether it's cytotoxic T cells or T helper cells, it really takes up to 2 weeks to slowly mature into a fully functional system. I skipped the natural killer cells. This is a weird cell, and we know very little about those cells in the chicken system. So if we look more closely at the gut is like the lung and like the reproductive tract, characterized by a single epithelial barrier covered by a mucous membrane, and underneath the cells of the immune system are sitting there. And what we know is that these cells are very closely communicating with each other. We always should keep that in mind. But these cells of the immune system, as we have learned over the last 10 to 15 years, in particular from work in mice, actually, and in cell culture experiments, that these cells of the immune system also very closely communicate with the epithelial cells. And epithelial cells have become a major focus in gut immunology research because these cells are a very critical component of the entire protection system. In mammals, we also have lymph nodes, and a lot of these cells can migrate to the lymph nodes and from the lymph nodes back to the mucosal tissue. They will form so-called memory cells, which are important in the context of vaccination, where we vaccinate and boost and generate memory cells, which upon challenge infection can respond very quickly to the challenge. And we will also have effector cells, those cells which can control infection, basically combat infection. In birds, we all know lymph nodes are lacking. We have up to now very little information what kind of replacement for lymph nodes we have in the bird. We assume that a lot of the functions going on in lymph nodes in mammals will take place in the cecal tonsil or maybe in the spleen and other organized lymphoid structures. And finally, and to make it much more complex, we have a microbiome. It has been mentioned already. So a whole range of different bacteria being in the gut. Sequencing has allowed to identify 500, even more, different bacteria in the as components of the microbiome of the chicken and also mammals. We still don't really know what is their function, and as we heard before, some new technologies like metabolomics may help us in the future to better understand how they communicate with each other, how they communicate with the epithelial cells and the immune system. So from work in mice, it has become clear that these bacteria in these commensal bacteria communicate with epithelial cells. Epithelial cells triggered by this communication may then communicate with the cells of the immune system. I showed you an example, but bacterial components may be directly sending signals from the microbiome all the way to the immune system and communicate with the immune system. So how to study this very complex system? What we basically did repeated work which has been done in mice, and it's much easier to do in mice than in chicken. But we were fortunate to be able to collaborate with colleagues in France who have a nice facility where they can raise sterile birds. So these birds are germ-free. We generated those germ-free birds. We took out different tissue samples, like from the cecal tonsil or just normal gut tissue. And then we applied a whole range of technologies such as immunohistology, qPCR, ELISA techniques, or more advanced holistic approaches like initially microarrays, but nowadays obviously RNA-seq, to better understand what is going on in these birds. But as we heard before, we have to compare the situation in these birds with a control group. And what we used as a control group initially were birds raised under SPF conditions. So that is not field studies. This is clearly laboratory work. And then we did these gene expression analysis. And what we found that there were a lot of genes upregulated in those birds raised under SPF conditions with some microbiome in comparison to the sterile birds. And when we look closer into these genes, we found that many of these genes, actually the number one function we found was immune function and development. And that nicely fits with what I've shown before, that there is a lot of developmental processes going on, colonization of these structures. And a large fraction of these genes were somehow related to the B lymphocytes. So I think it's clear to everybody that if we have IgY upregulated in SPF birds in comparison to these birds, this is related to B lymphocytes. The light chain of the immunoglobulin was different. The J chain, which is a junctional protein for IgA, was upregulated. Also, a couple of cytokines, which are really critical in development of B lymphocytes. And B cell markers. So it all pointed to some impact of the microbial colonization in B cell development in the gut. And in order to verify this, we did qPCR studies for, in this case, IgY, which is identical to IgG in mammals. And as you can see here, initially we did not see much of a difference. There isn't much of B cells in the gut, but within the 2, 3, or 4 weeks of development, we really found a highly significant difference between birds raised under germ-free conditions in comparison to birds raised under SPF conditions. And that was also true if you look here on the just on the right-hand side of the figure. That was also true for IgA production and likewise for the J chain expressed in the cecum. So this basically confirmed what we found with our holistic approaches, and then we wanted to see what does it look like in the gut. And if you look here, the germ-free birds, on day 7 we have very little B cells, almost no B cells. Day 21, no B cells. Day 28, no B cells, and even at day 55. And these studies were done in a layer type bird, I should say, but by day 55 a broiler would long be gone. So under germ-free conditions, they cannot develop a B cell system. While the SPF birds nicely started to develop a B cell system in the gut, as you can see here, and eventually formed these organized germinal center structures, thus indicating that they were fully competent of producing specific antibodies. We looked at IgA production, and as expected, no IgA at all in the gut, while SPF birds, as expected, would start to produce IgA around 3 weeks after hatch. What about macrophages? I initially told you that macrophages are there from hatch onwards. The system is already functional. And even though it looks as if there's a difference, we did lots and lots of cross sections, and basically there was not much of a difference between germ-free birds and SPF birds. As you can see here, they all developed or showed a fully mature macrophage system in the gut. So the microbiome did have a very strong impact on the B cell compartment, also on the T cell compartment. For sake of time, I don't show you these data, but I'll assure you that also T cell numbers were strongly reduced. But the innate immune system didn't seem to be affected very much. So we initially compared birds without bacteria with birds with bacteria, and what we wanted to know is, can we shift it from here to there by introducing a single bacterium? In this case, it was a probiotic E. coli, or by introducing 4 probiotic bacteria, the E. coli, Enterococcus, Clostridium, and Lactobacillus, and see what happens. And all these bacteria are nicely colonized, as you can see here. And then we compared initially germ-free to SPF as before, and you see the difference. So what happened in the case of addition of just one single E. coli? And there was not much in the context of B cells. 4 bacteria already started to do the job. You can see more and more B cells coming in. It's not yet at the same level as in the SPF birds with a more complex microbiome, but these 4 probiotic bacteria already help to develop the immune system. And this is confirmed if we look at immunoglobulin production. This is IgM and plasma, and you see here germ-free birds cannot essentially not produce IgM. Reconstitution with 4 bacteria helped a little bit. The SPF birds were clearly better and producing much more IgM in the plasma. IgY, it's pretty much the same picture. Germ-free birds do not produce IgY at sufficient amounts. It's a bit more in those with 4 bacteria, and it's significantly more in SPF birds. Same for IgA. So then we wanted to see the IgA concentration and compared plasma for IgA with cecal content. And you can see here also IgA was efficiently secreted into the gut, as we would hope to see, because it's doing its job in the gut lumen, and germ-free birds cannot produce any IgA in the gut, while reconstituted with 4 bacteria, it's produced some, and the SPF birds were much better. And also, these birds receiving just a single E. coli, or those receiving a cocktail of 4 bacteria with this particular E. coli, they could produce specific antibodies to these bacteria quite nicely, either IgA type or IgY type. So these birds, once they received a single bacterium, were actually able to respond to the single bacterium. So once we arrived at that point, we realized that our experiment was nice and interesting, but also a little bit stupid, because we used as a control SPF birds, as we scientists in the lab like to do. But that is not what is reflecting Mother Nature. So what we do under production conditions is this, and what the hen does is this. And the consequence under production condition would be this, and the consequence under natural conditions and the way it has been evolved during evolution is this, that these young chicks will pick up all kinds of environmental particles very shortly after hatch, and associated with these particles is the gut flora of the hen. And if you think about mammals, I mean, this is what happens in mammals during delivery. They also already get in touch with the maternal flora, so they get a starter flora for their gut, and that is not really the case in these birds, at least not a proper flora. So we added another group of birds where we basically gave little chickens at day of hatch some maternal shit from our hens. We put them in the hens on the bedding over the weekend. On Monday we had the hatch, and then we took out the hens and placed the newly hatched birds on this bedding containing maternal microbiome. And as you can see here, we call this group conventional. And if you compare this for IgM production, you can see here that even the SPF birds were quite lousy in comparison with those receiving a really complex flora. And that was also true for IgY and also true for IgA. So bottom line is we need more than 4 bacteria, and we need a complex microbiome in order to support proper development of the gut immune system. We repeated that with more birds, where we kept birds under SPF conditions and looked at plasma, IgY, and you can see here that is really going up significantly, at least 3-fold in concentration in the blood. And also in the cecum, you can see that in SPF birds, we initially found very little production, while we found nice production of IgA antibodies in the cecum. So clearly, the microbiome has an impact on the development of the immune system. And the real question is, how does it work? And this is still very much under investigation in mouse research, in human research, and what we have learned over the period of time, nicely reviewed in this review some time ago, that there are a lot of components like succinate, like lactate, like tryptophan and tryptophan metabolites, and also short chain fatty acids, which all have an impact on the cells of the immune system. And what is important to understand is that, let's have a look at tryptophan metabolites, that cells of the immune system have specific receptors called AHR on the surface, which can bind the metabolites and then signal into the cells to produce, for example, cytokines. And that is also true for the short chain fatty acids. And for the rest of my presentation, I will focus on that. Short chain fatty acids have been used a lot, and we heard that already before. What we know from research in mice is that macrophages have 3 distinct receptors for short chain fatty acids, which are called, these are G protein coupled receptors, and they have numbers. But they are also called free fatty acid receptors, 1, 2, 3. And we have a particular receptor which is called GPR109A. And these receptors are able to bind butyrate, propionate, and acetate, and maybe lactate. Others bind lactate. And they can then signal into the macrophage to produce some effector molecules. And what has been shown in mice is that this signal binding of these short chain fatty acids to these receptors and macrophages delivers an anti-inflammatory signal. But it also delivers a signal to the cells to tell them, please become an antimicrobial macrophage. So they limit inflammation, but at the same time, they are able to combat bacterial infections in particular, because this stimulates production of antimicrobial peptides, collectively called antimicrobial peptides. Some of them are defensins and the like. So this has been shown in mice, and the Jan Liljehoe's group at the USDA, in collaboration with her colleagues in South Korea, they nicely showed that this is also the case in the chicken. So butyrate will induce the production of antimicrobial peptides and macrophages. We were in particular interested in the effect of short chain fatty acids on the anti-inflammatory response. And what I should mention is that up to now, in the chicken genome, only one of those receptors has been found, which is the ortholog of the GPR43, or the free fatty acid receptor number 2. But that seems to be sufficient, because we established a method to isolate macrophages from the lamina propria. We were eventually, this is a procedure, I think I don't have to go through it, but we got a very nice population of macrophages, and they were not dead, but living. And so we could dump them into culture. And then in culture, we could culture them with medium alone, or we could culture them in the presence of butyrate. And what you can see is that these cells behave not differently. In the presence of butyrate or in the absence of butyrate, they produce very, very little of nitric oxide. Nitric oxide is a very important effector molecule produced by activated macrophages under inflammatory conditions. And you can see that here, if we added lipopolysaccharide, they produce huge amounts of this highly toxic metabolite. But if we had added butyrate to the medium, this inflammatory response was significantly inhibited. So butyrate in this system showed a nice anti-inflammatory effect on these tissue macrophages. They were from ileum. Here we have the same for macrophages isolated from cecum. And you can see here also a very nice reduction in the inflammatory response. Well, the inflammatory response is related to the production of cytokines, inflammatory cytokines. I have here just 2 of those. And we treated for 8 hours and 24 hours. And what you can see here, this is just butyrate. This is just, this is without anything. This is just butyrate. This is with LPS, and this is with both. And as you can see already, LPS is nicely inducing, again, production of interleukin-6. It's also nicely inducing the production of interleukin-8. And upon culture in the presence of butyrate, we see a nice reduction, and that is even more pronounced after 24 hours and highly significantly reduced. But this is really cytokine dependent. If you look at IL-6 and compare that with IL-8, you can see there is not much of a difference between those cultured in the absence or presence of butyrate. So in both cases, these cells were basically responding with the same IL-8 secretion pattern, while IL-6, a very strong inflammatory cytokine, was very significantly reduced in these birds. So short-chain fatty acids, not only in mice, but also in chickens, bind to a specific receptor, most likely. We haven't confirmed that, but that is most likely, and then can lead to an anti-inflammatory response. But there's also another very important cell, which is called the regulatory T cell. And regulatory T cells are critically important to maintain the tissue integrity in the gut. Without regulatory T cells, you get a lot of gut diseases. In particular, in mouse models, it has been shown that this leads to chronic intestinal inflammation and death of the birds. And this particular T cell population is absolutely critical. While over the last 20, 30 years, we have learned step by step a little bit more about all the other T cell subpopulations. We had we still have very little information about these cells. But they are critical because if you look at, for example, our salmonella infection, this will lead to inflammation and this will tip the balance towards inflammation. But what we really need at the end of the day is that this is eventually returned back to homeostatic conditions and the balance is tipped back. And this is achieved by the regulatory T cells to a large part through their secretion of 2 cytokines called interleukin-10 and TGF-beta. And as you can see here, T regulatory cells are the main producers of these cytokines. Well, these cells are not well understood in chickens, but over the last few years, we were able at least to discover the most important transcription factor in these cells, which is called FOXP3. And using that, we most recently were able to use RNA-seq and single-cell sequencing in order to identify the genes signature of these cells, and this hopefully will help us in future to sort out these cells and to better analyze and understand these chicken FOXP3 positive regulatory T cells and to investigate their role in the context of gut inflammation. And with this, I come to an end. I would in particular like to thank my colleagues at INRA Tour in France, Kathy Schuler and Philip Welch, for the beautiful collaboration with the germ-free chickens. You can imagine it's a really demanding technology, and the people in my lab, in particular Sonja Hertle and Sarah Lettmann, and those who gave us money to do the work. Thank you very much for listening.
Thank you very much, Professor Dr. Barn Kaspers, for such an outstanding presentation. You have nicely presented. There is no question in the chat box right now. From your presentation, Professor Dr. Barn Kasper, I have a question to you. What do you see as the biggest hurdle in successfully modulating the early life gut microbiome of chicks to build lifelong immunity against common mucosal pathogens?
Well, this is really a complex question. I mean, what has been shown is that a number of bacteria can control salmonella infection early on if properly delivered. Some probiotics. People in Belgium, Philip van Immerziel has nicely shown that there is a competitive exclusion mechanism where the bacteria compete with each other, maybe for nutrients. And in the context of active control, to be honest, these experiments are right now underway, that we have those technologies. And there is an impact on salmonella infection. Philippe Felch in France is working on that to see what is going on in the case of a salmonella enteritidis infection in germ-free birds or birds reconstituted with few bacteria in comparison to those with a complex maternal flora. But it's not all that easy, because we also know that during infection we see huge variability between individual birds. So in case of salmonella, we see high shedders and low shedders. So bacteria in some birds, even though they are from one line, I mean, one white leghorn line, for example, some birds shed a lot of bacteria, others can control it much better. And that is maybe due to competitive exclusion, but I don't know. But I would say that a proper colonization early on will definitely help. It's one aspect in the whole system will definitely help to improve gut health.
Thank you, Professor Dr. Ban Kasper, for highlighting the answer. Now it's over to Professor Dr. Mohammad Kashif Salimi. Actually, we were not joined by some of our keynote speakers. I think one of our keynote speaker, Professor Dr. Mark Purcell from University of Delaware, USA, and oral presentations for Professor Anjum from the University of Bristol, UK. So I would like to ask Professor Dr. Kashif Salimi to please end up this session with some concluding remarks to our keynote speakers and to the participants who are joining this conference.
Okay, you, thank Professor. You very much, Syam. Am I audible?
Yeah.
Okay, thank you very much for our speakers, keynote speakers, especially Professor Rob. And our apology that there is a huge time difference between Pakistan time and US and Canada time, and also European time, and they have spared the time. And then I'm really thankful to the Professor Professor Korer from the Canada. I think he has also experience of visiting Pakistan, and Professor Bernard Kasper, he has also frequently visiting international conferences and meetings, and he was in the Lahore in Nishat Hotel. There was an academy organized by the Boehringer Ingelheim in Pakistan, and I was also among the speaker with Professor Kasper. So this is always really delightful to listen these high-level speakers, because they are having a experience in their field, and I can say when we are proud that we are having time with these people. So thank you very much, Professor Casper, Professor Korar, and Professor Anoop for sparing your valuable time. Now it is over to Sam. Sam, please continue for presentation of certificates.
Yeah. Thank you, Professor Dr. Kashif Slimi. As a token of our appreciation, we would like to deliver certificates to our keynote speakers. Please join me in delivering the certificates to our keynote speakers. So the first certificate is to Professor Dr. Doc Hoover from the University of Alberta, Canada.
I think Professor Korver is with us.
Yeah.
Yes.
Okay, Professor, thank you very much. And this is a certificate from our side, from Poultry Professional Society, certificate of appreciation for sparing your valuable time and your commitment to this event. And we really learned a lot from you. Thank you very much.
Thank you. My pleasure.
Okay. So the next certificate goes to Professor Dr. Ban Caspers for his valuable time.
Bernd
Professor
Casper.
Kashif Slimi.
Thank you.
Yeah.
Professor
very much.
Bernard Casper. Thank you,
I greatly
Professor, for
appreciate.
Professors,
Go
over
ahead.
to you.
Yeah, thank you very
Yeah.
much for inviting me. It's
Thank you very
always
much.
a great pleasure to talk about the chicken immune system. And I also have very nice memories
And I
of
think,
my visit to Lahore a couple of years
I
ago,
think this,
which was really
I
great.
think this was very important topic because yeah, Professor, I was talking that this is a very important topic, always chicken immune system, because nowadays chicken immune system is central thing that control the diseases, and we are facing the challenges of avian influenza and other diseases. So I think this is very important to learn from you.
Yeah, thank you very much again. I greatly
Thank you.
appreciate.
Okay, our next certificate of appreciation for Professor Anoop. He was our first speaker today. And thank you very much, Professor, for sparing, if you are with us, for sparing your valuable time and your commitment. And I really we apologize that that was truly odd hour when you presented. That was almost 3 a.m. in US. So thank you very much, Professor. And I hope this collaboration will continue.
So with this, Professor Dr. Kashif Slimi, I would like to ask you to please conclude with some remarks.
Okay, thank you, Sam, for the official closing of this session. I'm really thankful to all our three speakers and the team, really led by Mohammed Siam. That was a wonderful team that organized these events, and especially this is a second very successful graduate research conference. And Siam, you and your team really did a wonderful job. Thank you very much.
Thank you very much, Professor Dr. Kashif Slimi. So now we end up with our two scientific sessions. For the first session was poultry nutrition and alternative feed resources, and the second was poultry diseases and gut health management. We now have scientific session number 3 on poultry genetics and genomics, and that session will be chaired by Professor Dr. Sami Dalidi from the University of Arkansas, USA. So this session will begin at 3 p.m. in Pakistan time. So between that time, we have a break for coffee or for tea. So please join us after 30 minutes. Thank you very much.